Hello and welcome to the Drug Science Podcast with me, David Nutt. Here we're bringing together experts and activists for a rational, honest and informed conversation about drugs.
A Fascinate Productions podcast for drug science.
So welcome to this Drug Science Podcast. I'm delighted to say that tonight I am speaking with Dennis McKenna, who is one of the world's leading, maybe the world's leading, ethnopharmacologist. He's joined me from British Columbia in Canada, and he's going to share with us his 50 years experience of working in this field. And I think you'll find it fascinating. Welcome, Dennis.
Thank you, Dr. Nutt. It's a pleasure to be here. I've been following your work for a while, and I very much admire you and what you've been doing. You're a change maker. Thank you for the nice words. I don't know if I'm the most foremost ethnopharmacologist, but I guess I can legitimately claim to be a ethnopharmacologist, and we could get into whatever that means.
Well, that's a great place to start, Dennis. What is an ethnopharmacologist?
Well, there's a kind of a tortured definition of it, but it kind of makes sense. Ethnopharmacology is the interdisciplinary scientific investigation of natural biologically active substances used or observed by humans in indigenous societies. And that's a many-part definition, but it kind of covers the waterfront in the sense that, you know, if we didn't restrict it to indigenous or traditional societies, then everything we do with respect to pharmacology would be ethno, because, you know, we're humans doing this. So that's one point, that the focus is on traditional or indigenous use, not modern use. Mm-hmm. The other thing is that, you know, ethnopharmacology is not necessarily confined to medicines, nor is it confined to plants or fungi or any of that. It can be from any source, and it's not necessarily about biologically active substances that people ingest. You know, for example, arrow poisons is totally a legitimate area of inquiry for ethnopharmacology. So it has to do with humans in traditional societies using biologically active substances for medicines, poisons, even foods, nutritional, and that kind of thing. And that's why it's such a tortured definition. And that comes from Bo Homestead, who you may be familiar with, or maybe not. He was, you know, the old school. Mm-hmm. And I lifted that definition from him.
It's a complicated definition. Maybe you should help us by start telling us how you got into this and what your pathway has been, because I gather you've been doing it for about 50 years.
Yes, I have, actually. I grew up in the 60s and was a teenager during those years when the counterculture was on the rise and everyone was excited about LSD and other psychedelics, but LSD was kind of the focus of the attention. My brother, Terrance, also well known, he was 4 years older than me, and he was in Berkeley while I was stuck in Colorado in high school, but he was a major influence. We were both fascinated with psychedelics. Not so much LSD. What we were fascinated by was DMT, which was very rare in those days, but seemed to be an order of magnitude weirder and more interesting in some ways than LSD. But that's another story. And at the age of 18, I encountered two books, one of which was the first edition of Carlos Castaneda's book, The Teachings of Don Juan, which my brother gave to me for my 18th birthday. And even though I think there's a consensus that that book is, much of it is fiction, or at least certainly loosely associated with the truth, and yet for me, I didn't know that at the time. And what that book gave to me was a perspective that there is an ethnographic and historical context for psychedelics. Mm-hmm. And the other book that came onto my radar at the same time, or about the same time, I'm not sure how I acquired it, but it was a proceedings of a symposium that was held in 1967, sponsored by the National Institute of Mental Health. It was called the Ethnopharmacologic Search for Psychoactive Drugs. And it was a private conference, even though government sponsored, but it was a closed conference, but that book was issued. So those two things, those two books really influenced me to go into this profession. And then actually 50 years later, in 2017, I staged a symposium, 50th anniversary symposium of that original symposium, and in England at Tyringham Hall. And we published the proceedings of the 2017 symposium, and also we reprinted the 1967 symposium. The 2017 symposium, because of availability of live streaming and all the technology that we have now, that was watched by 75,000 people. So I was very happy that a lot more people in the world know what ethnopharmacology means now. Yeah.
50 years is a great time to reflect backwards. I mean, what have been the major changes you've seen in the 50 years, apart from greater interest?
Amazing changes, and even discoveries that were not really imagined in the 67 conference. Originally, the idea with that conference was that there was going to be follow-up symposia every 10 years because ethnopharmacology is an active field. Well, the war on drugs came along. The U.S. government became embarrassed that they had anything to do with this. So it never happened. And I just felt like within 50 years there had been significant discoveries, and we wanted to address those. What was discussed in the 2017 symposium, we had sections on kratom, which is not exactly a psychedelic, but it's a very interesting plant. We had maybe too much on ayahuasca. Well, we had excellent, ayahuasca was not unknown in the 70s, but we know a lot more about what its chemistry was. It was a significant review of the progress in the last 50 years. So I was happy to be able to contribute to that and play a role in organizing that. I think it's raised awareness of the field.
You're an 18-year-old. You've got this book from your brother, and you've read this conference proceedings. Yes. What did you do then? Did you go off to university to study this, or how did
yes,
you? Yes.
Well, after that, at 18, then I went into undergraduate. I was an undergraduate at the University of Colorado, and I majored in what they call distributed studies. What I wanted to do was put together a program similar to ethnopharmacology or ethnobotany. Well, there was no such thing, so I decided to study botany and chemistry and anthropology, and eventually I ended up taking quite a lot of courses in philosophy of science as well, which I never regretted. I felt like that was very useful to my education. So then I ended up going to the University of Hawaii for my master's degree. I got a forestry grant from the Department of Forestry in Hawaii, and they said, well, we don't care what you study as long as it's Hawaiian and as long as it's a tree. And I thought, great, I'll study acacias, because 2 or 3 important tree species in Hawaii are acacias, and acacias are well known for their content of tryptamines. There are many, many species in that genus that contain significant amounts of tryptamines. I thought I would study the alkaloids of the Hawaiian acacias and be, kind of a phytochemical survey. Turns out the Hawaiian acacias don't make any alkaloids. So I ended up studying something. I ended up studying acacias, but I studied non-protein amino acids, which were also interesting as sort of biochemical markers. Then when I finished my master's at University of Hawaii, I ended up coming to the University of British Columbia for my graduate work. And I was originally going to work on psilocybin. I did for about a year. I worked on, the objective was to study the enzymes and the genetic regulation of psilocybin biosynthesis. To be honest, I was failing fungal genetics, which is an incredibly complicated field. And then my supervisor, Neil Towers, kind of threw me a life rope. He said, well, maybe you'd like to go to Peru. I have some extra money in the grant. So I said, well, my bags are packed. When do we go? And so I shifted my whole focus to ayahuasca. So that was the direction I ended up going in, looking at both the pharmacology and the many admixture plants used with ayahuasca. It's a combination of plants containing MAO inhibitors, namely beta-carbolines, other plants containing DMT. And of course, the beta-carbolines protect the DMT, and so it's orally active. And that was the whole rationale, was to explore this mechanism of oral activity. Ayahuasca is, of course, world famous. Everyone's heard of it. But there was another group of indigenous psychedelics derived from Varrola, which is a genus of trees that has sap very, very high in the tryptamines. And it's used throughout the Amazon as a snuff because of this oral activity.
Oh, yeah.
There was a Witoto preparation called Kouhei that was orally active. And I wanted to compare the Kouhei with ayahuasca, completely different botanical sources, but similar chemistry and similar mechanisms of action. So that's what my thesis ended up being about.
But sorry, just to be clear about the tree-derived, does it contain its own monoamine oxidase inhibitor? I wasn't familiar with this.
Well, that was the question.
Or do you have to mix it?
Yeah, that was the question. The mechanism of action for ayahuasca was pretty clear. Although I did do the bioassays, I did the in vitro assays and so on, and validated that was what was going on. And then the idea was, with the Kouhei, are there beta-carbolines in there, or are there something else that is inhibiting MAO? I never really did get that figured out. But what I did find out is that essentially the tryptamines were self-potentiating. The extracts usually contained combinations of DMT and 5-methoxy-DMT. And if you eat enough 5-methoxy-DMT, it doesn't really require an MAO inhibitor. And in my in my bioassay work, I found if I took these extracts and removed the tryptamines, then it lost MAO activity. So the best I could figure out is that it was the tryptamines that were kind of self-potentiating themselves in that preparation.
I see.
And another aspect of that work is that this was dying knowledge. The curanderos that we interfaced with, they were happy to prepare sample for us, various samples, but they all pretty much have said, this is something my grandfathers did. This is something my fathers did. I don't really remember how they did it, but I'll give it a shot.
I mean, how hard was it to get funding in those days?
About 10 years after I got my degree, I got an opportunity to participate in a biomedical study of ayahuasca that was initiated by one of the Brazilian churches, the UDV. For that particular project, I was able to get funding from private sources, partly through the Hefter Research Institute, which was just starting at that time. And then my brother had started a nonprofit called Botanical Dimensions. And the idea of Botanical Dimensions was to put a gene bank, essentially an ethnobotanical garden of these psychoactive species in Hawaii. And through those channels, I was able to get funding for this project.
But you were in a university, were you? You were developing an academic career in this rather weird topic. That must have been challenging. I waited till I had an academic career before I started.
Yeah, it was definitely challenging because here I'd worked, if you count the 8 years, or if you count the master's degree in Hawaii and the work in British Columbia, 8 years altogether to get my PhD. And then I come out with this PhD in this not only interdisciplinary but quite controversial area. Not a great job move, not a great career move if I'm looking for a job. And I was, I finished my PhD and I went back to the States because, the Canadian government said, well, now that we've subsidized your education, you have to go back to your third world country and help your people, So I went back and I started doing what seemed like an endless series of postdocs. I ended up working on selenium as an anti-cancer agent, and the only part of my skills that were relevant was the fact that I knew sterile technique and I knew how to grow fungi in culture. And that wasn't because of anything I learned in school. That was because I taught myself how to grow mushrooms. But after that, I ended up going to National Institute of Mental Health. I got a fellowship in the Laboratory of Clinical Pharmacology.
So that was in Bethesda, in Maryland? In
Bethesda, Maryland, yeah. I spent 2 years working under Dr. Juan Saavedra. He worked under Julius Axelrod. He and Axelrod were the first people to study endogenous DMT. They reported on the occurrence of DMT in rabbit's lung, and I was familiar with that work. So here I am working at my first postdoc in San Diego. And, in those days, they used to request, if you wanted a paper, you sent a postcard, pre-electronic.
Yeah, I remember
those
days.
A postcard requesting my publications about ayahuasca from Dr. Juan Saavedra. I wrote back and I said, I'm familiar with your work. Is there any, and I'm very interested in this area, is there any way I could potentially come work with you? And he wrote back a very nice letter, and he said, well, there is a program called the Pharmacology Research Associate Traineeship, or PRAT Fellowship, and it's for people who are not specialists in pharmacology or neuroscience, but it's a way to get them an entry point. And I was accepted into the program. After I did that postdoc, I ended up working with Steve Peruka at Stanford. And at that time, the MDMA neurotoxicity issue was very active, and people were looking into that, and Dr. Peruka was investigating that. And one of the reasons that he decided to leave Stanford and go work for Genentech is that he was not awarded tenure over a controversial thing that happened, which he did a survey of Stanford undergraduates about their uses of MDMA. And it was connection with the concern about neurotoxicity and so on. And he reported that about 30% of undergraduates at Stanford were using MDMA or had used it. That was really not the politically correct thing to do. And you're familiar yourself with what happens when you come a cropper with the powers that be. So Steve said, I'm not going to be here. You've got to find something else to do. It's about time you look for a real job. And at the same time, about that time, there was a new company starting up in the Bay Area called Shaman Pharmaceuticals. And the idea with Shaman's mission was ethnobotany-driven drug discovery. And I interviewed, and and my job title was Director of Ethnopharmacology. I just had completed these 2 postdocs at Stanford and NIH, so I was armed with a lot of the techniques for receptor binding and all that. So I developed their receptor lab. And we were screening plant extracts for all sorts of activity, but primarily analgesic activity.
It's quite a transition from working with traditional plants and traditional peoples to more what you might call classic neuropharmacological screening for commercial purposes. That must have been an interesting challenge for you, I suppose at least you saw both sides of the coin.
I had these skills which were very, very fresh and kind of cutting edge of what was the state of the art at the time that could be applied to the screening of these plant extracts. So that's what we did. And we screened a bunch of things. One of the plants that we screened was Salvia divinorum. The chemistry of that, of course, they're very select. The salvinorin A is extremely selective kappa opiate agonist, and I was running these extracts through all the known opiate receptors. We had assays set up for all those things, and I just kept scratching my head. I mean, I was getting incredible activity at all of the opiate receptors, and I thought, this can't be right. It can't be this potent. It was not understood what it was at the time, what the pharmacology was. But then years later, Brian Roth worked it all out. But I reported my results on these binding assays to the upstairs management, my supervisors in what amounted to the pharmacology department. And they said, well, it doesn't matter anyway because we don't want to discover another morphine. Well, of course, it wasn't. But anyway, we won't get into that.
No, let's not go there. It's been a hard. The kappa receptor's been a very tough one to turn into medicine. So you're probably best to give up on it.
Very difficult, yeah. So I worked there for about a year and a half, and I was offered a job at Aveda, which is a cosmetics company. And they were interested in new ingredients from the rainforest to put into their cosmetics. So they sent me to Brazil, to look for some of these plants. While all this was going on, I was developing this plot to go down there and do this study with the UDV. So when I was supposed to be in the rainforest collecting plants, I was actually in Manaus running this study with my colleagues. Many publications resulted from that. We did some interesting work, and some of it, really still needs to be followed up. One of the questions that we naively ask in this project is there any biochemical marker? Is there anything that makes ayahuasca drinkers different than ayahuasca non-drinkers? Is there some aspect of their neuropharmacology or anything? We asked a very vague question. But something came up as we evaluated the samples. When we did the study, we took blood samples. We were evaluating platelets. As you know, platelets are often a peripheral marker of what's going on in the central nervous system. And we found this anomalous result, which is that the drinkers of ayahuasca had an elevated titer, an elevated abundance of the serotonin transporters. And it was a persistent thing. It wasn't an artifact of taking ayahuasca. These were subjects that were, they went through a washout period, so it wasn't a residual effect. It was apparently a persistent, elevated upregulation of the serotonin transporters. And we sort of thought, well, what does that mean? That's interesting. But what does it mean? And it actually didn't take too long to find out. There was a whole literature about pathological deficits in the transporters associated with things like suicidality and depression and even homicidal behavior, addictive behavior, all of these things. There's I don't know how current this work is, but at that time, deficits in the transporters, not only serotonin but also the other monoamines, was being linked to these pathologies. So we thought, well, that's very interesting that ayahuasca apparently reverses this, because in our psychological screening that we did of these subjects, it was a textbook case. I mean, it was almost too good to believe, because all of the subjects in our UDV study were people who had joined the UDV in a state of life crisis. And it usually had to do with addiction, alcohol addiction primarily, and other kinds of just dysfunctionalities like depression, like suicidal ideation and this kind of thought. They all pretty much told the same stories. We did extensive life story interviews with them, structured psychiatric life story interviews. My colleague, Dr. Grobe, was working on this project. He was the PI.
Yeah, yeah.
And they all told the same story, that they came to joined the UDV, usually because a friend had recommended it, and they were in a state of life crisis, and they felt that their experiences were not only terrifying initially, but redemptive, in the sense that they said they could see the way they were headed. They could see the direction of their life, and it was like a big wake-up call that you have to change. And they took that lesson to heart. Most of the people, or at least all the ones that we interviewed, have been at least 10 years in the UDV, and some have been in the sect for 30 or 40 years. And they all felt that it was two things that kept them on track. One was the medicine, the tea, as they called it, and the other one was the supportive environment of the UDV. It's a very nurturing kind of religion. I used to kid them. I used to say, you guys are like psychedelic Mormons or something. You're, in the sense that Mormons are also a very community-oriented kind of religion, and that's what the UDV was.
But isn't that what psychedelics do to some extent? Don't they bond people to others and to the world?
Indeed. Well, I think psychedelics play roles in a lot of these religions that they haven't really acknowledged. Even Christianity, there's good evidence, or certainly suggestive evidence, that mushrooms may have played a role in Christianity. I mean, there's some very peculiar iconography in some of these stained glass windows and this sort of thing. So that's a whole area to be explored. Not by me.
Let's fast forward now, 30 years. So
okay.
What are you working on at present?
Well, right now, so I did that work, and then I since I couldn't get a job in academia, I became more or less a consultant for the natural products industry. Then my brother got sick in 1999, and with terminal brain cancer, with glioblastoma. I basically dropped everything, and I devoted that year to being there for him. And after he passed on, there was nothing. I was broke. I couldn't do consulting anymore because, when you're a consultant, if you don't do it, it dries up. And then, again, another lucky break. I was invited to join the Center for Spirituality and Healing at the University of Minnesota. I used to tell people it's not a New Age cult. It's actually, it just sounds like one. It's actually the complementary medicine program. And they wanted someone to teach ethnobotany. I started in 2000, and I terminated it in 2017. So I ended up teaching ethnopharmacology, another course called botanical medicines in healthcare, and another lovely field course every January taught in Hawaii called Plants and Human Affairs. And that was great. I really enjoyed that, and I continue to do that.
Especially in January.
Not a bad trip to take if you live in Minnesota and want to go to...
Good call, that one.
Right, it was an intercession course. So I taught that for several years and really enjoyed that. And then maybe this is worth mentioning. In 2004, I got a grant from the Stanley Medical Research Stanley Medical Research Institute. And they were interested in natural products. They were looking for potentially new medications to treat positive symptoms of schizophrenia. They contacted me and said, well, we're looking into Chinese traditional medicines as potentially new medications for schizophrenia, and we have nothing going on in South America. Would you potentially be interested in applying for a grant? And I said, well, sure, why not? That was a great project. We didn't find the medication for schizophrenia, but what it did let me do was renew all my connections in Peru that I had from years before. and we collected about 150 species of plants that basically had ethnomedical uses that looked like they might be psychoactive or even specific for mental illness. The plants that sort of rose to the surface in this screening project, they were not novel compounds. They were from plants in the Apocinaceae family that had been looked at to some extent in terms of their phytochemistry. So they contain these complex indole alkaloids, but we didn't have the bioassays to follow up, and so that's where the work has sort of, I wouldn't say stopped, but it hasn't been advanced.
Well, it may be that the rising interest in, I mean, a whole slew of psychedelically oriented companies being set up. Maybe they could broaden their remit a bit and get back into some ethnobotany.
I would like to. And actually, British Columbia seems to be a very fertile ground for incubating these various psychedelic-oriented startup companies. And, I mean, they're cropping up like mushrooms all over the place. They all have, a lot of them have an orientation to mushrooms. It's almost, and that's great. I think psilocybin is just about the perfect clinical psychedelic, but it's not the only one in the universe. And there's still great potential, I think, to find new psychedelics in nature that are, we think of sort of the top pantheon, ayahuasca, mushrooms, iboga, and so on. But there are undiscovered psychedelics out there, I'm convinced, and would be worth going after.
Yeah, and it may be the commercial imperative, the ability to get something patented.
Exactly. And a lot of these companies, psilocybin, it's hard to patent psilocybin. It's been in the public domain for so long. You might patent something about the synthesis of it. A lot of companies are looking at formulations, and I think this whole interest in microdosing is, in some ways, it's a way to try to come up with a patentable formulation. But I have been talking to several of these companies, and I'm saying if you would consider investing in drug discovery, we can find new psychedelics that nobody knows about, structurally novel psychedelics.
Well, good for you, yeah, and I hope they're going to take you on as a consultant.
Yes. I hope something comes of it. What I would like to do is leverage my connections at the university in Iquitos, Peru, the National University of the Amazon. All of those connections going back to 1981, those folks are still there, and the infrastructure for collecting plants and all that is there. It needs to be married with the capability to do chemistry and do, pharmacognosy and phytochemistry on these things.
You're the man to do it, Dennis. You're the man to bring them together. There's no doubt about that.
Well, I hope so, but I don't know. I'm not getting any younger, David. That's the problem. I feel like there's so much to do, so much that I still want to do.
Well, one of the things I hear you've been doing is you've been developing some tribute to your brother. Tell us a bit about that, and tell us also about how you his very interesting insights and what you think of them and how they influence your thinking.
Yes, we've been doing a tribute to Terrance that's been a virtual event. It's connected with a nonprofit that I founded about a year ago called the McKenna Academy of Natural Philosophy. The idea is to create a platform for psychedelic education, either online or maybe eventually a physical location in South America. That is an aspiration. I don't know if it'll ever happen. But in connection with the Academy, we were going to do 2 big symposia in San Francisco at the beginning of April. Then everything got canceled. So the 3rd of April was the 20th anniversary of my brother's passing. And so we had this idea. So we decided to pivot and do it online, which we did. We did actually 4 installments, and basically me talking to old friends of Terrance. I'll send you all the links.
That's very helpful. If you could, I'll promote them. Absolutely.
Absolutely.
Because I will tweet about this and we'll
they're
promote
open
the links.
access. They're free. Anyone can look at those things. And it turned out to be, we had 8,500 people sign up for this series. I'll send you a link to the website and send you these other links, and you can put those out if you like. I would really appreciate it.
Please do that. Yes. Before we stop, I've got to talk to you about this La Charrera experience, which you've talked about. Could you like to share that with us?
Well, yeah, the La Charrera experience is it's so hard to explain. It's hard really to describe what went on. It's all discussed in great detail in my memoir, the Brotherhood of the Screaming Abyss. but roughly, so here's the backstory to that. As I mentioned earlier, my brother and I were obsessed with DMT back in the 60s. And we felt that it was just an order of magnitude more interesting than LSD. But DMT was frustrating because you can only spend about 10 minutes, in that place, and you don't come back with much except a sense of something really astonishing just happened. So we were looking for something that would prolong the experience. And that's why we got interested in this Witoto drug. We didn't know at the time, nobody knew around 1971 that that's what ayahuasca was. Ayahuasca was basically a orally active form of DMT. The role of the admixture plants was not well understood. But so we went for this Kouhei, and that's what took us to La Charrera, because that was the ancestral home of the Witoto. Well, when we got to La Charrera, we had been cautioned by an anthropologist that we had encountered on the way, and he said, you can't just go in there and start talking about Kouhei. This is like big shamanic magic. They will not be happy for these crazy hippies to and we definitely looked the part at the time, to wander in this village and start talking about Kouhei. You need to be very careful, We thought, okay, all right, we will be careful. But so when we got to La Charrera, it turned out that it was a rainy season and there had been pasture cleared around this little mission village of La Charrera, maybe 200 acres or so of pastures with cattle and mushrooms growing out of every cowpie, basically. So we knew what they were. We had absolutely no experience with them, but we knew from our homework what they were, and we kind of had a very cavalier attitude toward them. We thought, well, this will be great. We can enjoy these while we're waiting for the real secret to show up. Well, the mushrooms very quickly made it clear that they were the real secret. And they were what we'd come for. And in fact, if you think about it, psilocin is really DMT in an orally active form. I mean, it is the perfect orally active form of DMT.
It's a prodrug, isn't it?
Yeah. And we started eating these things in a recreational or fairly thoughtless manner because they seemed so friendly. And then they started downloading information that was really out of left field and instructions how to do what, I'm not sure, but essentially turn ourselves into a UFO. I'm not sure. Well, that didn't work, but that was the experiment at La Charrera. Were you at Breaking Convention in 2017? Were you there?
Yeah, I was there. Yes, yeah. But it was so huge and so many things going on in parallel. Yeah, in fact, I mean, I knew you were there, but I didn't see you. I mean, it's.
I gave a talk. There called "The Experiment at La Charrera: Psychotic Break, Shamanic Initiation, or Alien Encounter." And I was kind of arguing that maybe this was an alien encounter. I mean, we certainly thought it was at the time.
It is interesting, isn't it? Why DMT doesn't leave these more enduring sense of change and understanding and insight that LSD does. You think it is because it just shows short acting?
It's short acting, yeah. Which is one reason I think these experiments going on now. I think with Robin Carhart-Harris and Chris Timmerman on the extended DMT state, which you're involved with, right? You're
yes, that's true. I'm involved with it. I'm trying to make sure the UFOs don't steal them away.
Yeah, we have to be careful about these things.
Indeed. These are powerful drugs, and it's
definitely.
I mean, the field has moved on a lot, but there's still. I keep telling everyone that just because there's not a lot of public criticism of what we're doing doesn't mean that the public is supporting, and certainly not some of the. There are aspects of the media, I'm sure, who would be delighted for something to go wrong, one of our experimental subjects to have a bad response, and they would then swing the pendulum back and try to crush it all. So it's still in the infancy, the psychedelic renaissance.
But as a pharmacologist, David, my question for you is it really necessary to extend the DMT state, or does psilocin do that? I mean, at high doses, the phenomenology of psilocin is very much like DMT.
I mean, the reality is that there are commercial interests in trying to do that because psilocybin, as you pointed out, is very hard to get any kind of patents, and so people are seeing DMT as an alternative, and maybe a slow release or a slow infusion of DMT could be commercially viable. I mean, that's the driver. I think from my perspective as a neuroscientist, the work that Chris has done already at Timmermans is. I'm actually going to have a podcast on that shortly, but that's shown quite clearly that the fragmentation of certainly EEG fragmentation, you see desynchrony with DMT is profound, but very similar to that with LSD and with psilocybin.
Right.
The 2A receptor does seem to do the same thing, regardless of what agonist stimulates it, which of course is what you predict as a pharmacologist.
And it seems that. Yeah, I mean, I'm all for the research. I think we should support the research, even if it doesn't result in the eventual clinical application. It's fundamental to understanding the nature of consciousness.
I totally agree with you.
That's really where these medicines are. That's their promise, So I'm in favor of that, and I'm not sure this will ever result in a clinical application, but it doesn't really matter. It's worth doing.
Exactly. And actually, what's fascinating about one of the people might say, well, why study Amazonian or Brazilian churches that use, strange psychedelic drinks? But in fact, the reality is those studies have given people like me a lot of confidence that regular use of these substances is certainly safe, and even if it's not necessarily, you can't prove it's beneficial, at least. And that was quite influential when we were getting permission from our government, our regulators, to do the first psilocybin imaging study.
Oh yeah.
We argue that the historic experience shows these are safe, generally safe, and that helps sway them because, of course, to do the full toxicology that normally you'd have to do for a pharmaceutical was way outside our budget.
Right. Well, the centuries of indigenous use testifies to the fact that they're at least not toxic, and when we try to integrate them into a clinical context, maybe there are some issues, but really the significance of the UDV study that we did in the early 90s was not only did it raise questions about this persistent upregulation of the 5-HT transporters, which I'd be interested to hear if that has any significance, but that seemed to be a marker that was directly correlated to the reversal of these deficits. I don't say it's causative.
No, I mean, it's been reported in other conditions. That it seems when people fall in love, those sites upregulate as well. So it may be a marker of a sense of tranquility or well-being or of harmony. I mean, of course, the challenge is how would a platelet, which comes out of the marrow, how would that change in relationship to your subjective state? And it's not implausible. It's driving it. I mean, it's not obviously how it could drive central effects, but it's not implausible because it may be that whatever causes the upregulation in the periphery could cause the upregulation in the brain.
Well, it's a central nervous system effect. We were able to establish that. I mean, we were looking at platelets for screening them against a number of receptors because that's a peripheral marker, supposedly, or at least at the time, of changes in the central nervous system. But then one of the people involved in the study, Jace Calloway, for various reasons, we thought that the agent that was causing this prolonged elevation was probably tetrahydroharmine, one of the ayahuasca alkaloids. And he returned to his lab. He had access to, I think, SPECT imaging technology in his lab.
Yep.
So he tried it out on himself. He started taking tetrahydroharmine, and then he could actually do SPECT scans on his own brain and trace, measure this elevation in the transporters. And then when he ceased taking it, it faded away after a couple of weeks. And so it was a transient effect. But interestingly, the UDV, they take it about every 2 weeks. So maybe in the use, maybe intuitively they understand you need to top up the tanks about every 2 weeks.
And that's what's very interesting, because one of the big challenges for us, Dennis, in terms of our depression studies, is we get powerful effects from a single strong psilocybin trip. But in the majority of people, this wears off over a period of weeks or months. It will be really wonderful to find some kind of way of maintaining that, and maybe a fortnightly dose of ayahuasca would be a sensible way forward.
Yeah, or even potentially it doesn't have to be ayahuasca. It could be, I think it'd be very interesting to do a study with just tetrahydroharmine, which you wouldn't perceive much of a psychoactive effect, but with the imaging tools that you have available, you could actually take a closer look at this, because we never really looked into this anymore. I mean, we reported the effect, and there's been no follow-up as far as I know. So if you have somebody interested, I think this would be a good area to explore.
Again, you'd run into the same problem if the ethics committee would say, well, what's the safety of tetrahydroharmone, but you could argue that if it's something that's been used for centuries in indigenous Brazilian populations, it probably is safe. Do you have any idea what the dose is? Send me the info, because we can talk about the dosing, and let's not get too technical, because we've talked for a very long time, and I just want you to reflect just for the last few minutes about how you must be pleased to see that your home state, well, at least Denver, is allowing the mushrooms to grow again, and Oakland. Are you pleased by these movements to sort of liberate plants for human use?
Yes. Yeah, I am pleased by that, particularly these decriminalization movements, are a very hopeful sign. For one thing, the very idea that any of these plants should ever be criminalized is absurd. I mean, you can't criminalize a plant. They are not criminals. The people who criminalize them may be criminals, but the plants are. So we need to, make that conversation clear. And like everything, it's complicated. I mean, the Native American Church has just issued a statement saying, we don't want to be part of this. We don't want you to say peyote is one of the ones that should be decriminalized, because
really,
it's protecting. Criminalization was protecting it, and it's under a lot of environmental pressure due to overharvesting.
Oh, I see. Yes, of course.
Same issues result from ayahuasca. I say that is fine. That's worth a conversation, but that we should put forth the notion that no plant is criminal, and that people have the right to form symbiotic relationships with plants.
Dennis, it's been wonderful talking to you. We could have talked for a lot more, and maybe at some point we'll get you back.
Thank you.
But thanks very much indeed.
I would love to come back. It's been a real pleasure. Yeah,
great.
Let's keep in touch.
Let's absolutely do that.
All right. Thank you so much.
Bye-bye.
Bye-bye.
I hope you didn't mind two old men reminiscing about pharmacology, plants, science, etc. It's one of a whole series of podcasts which Drug Science is putting out. Follow me on Twitter, Prof. David Nutt. Follow Drug Science on Twitter. And hopefully, if you enjoy what we're doing, join the drug science community and support our initiatives, both in education and in policy change, by becoming one of us. Thank you for listening.